Hmm, have you ever wondered? Hi, I'm Kalpa Tinka and Kobe by night, and you're watching Chem Simplified. We've already heard about the basic facts about lipids, their nomenclature, and some saponifiable lipids. I'd like to continue our discussion with Part 6 of lipids, glycolipids, and non-saponifiable lipids. But first, can you guess these shuffled words right? I'll give you 5 seconds to answer each. Let's go! If you guessed these words right, congratulations! We will be dealing with these words today, and hopefully by the end of this lesson, we may be able to identify and illustrate structures of glycolipids and non-saponifiable lipids and name them appropriately, to differentiate and identify the various functions of glycolipids and non-saponifiable lipids, to appreciate the functions of glycolipids and non-saponifiable lipids and apply them to real-life situations, and to relate the use of glycolipids and non-saponifiable lipids by showcasing examples of their uses. Alright, now that we're done with our preliminaries, I would like to introduce you to my friends, the glycolipids. Yes, I'm lonely, but do any of you have an idea what my friends are? Well, glycolipids are a class of lipids containing carbohydrate residues and are usually the major lipids of bacterial and fungal walls. They function for the stability of the cell membrane, where it is also important for cell recognition and connecting cells to form tissues. Now, search up, because I have another question. What is the parent compound for glycolipids? That's right, ceramides. They are the long-chain amino alcohol called sphingosine, to which a long-chain fatty acid is joined by an amide bond to the nitrogen atom on carbon 2 of a sphingoside. The glycosidic bond formed between the primary alcohol group of the ceramide and a sugar residue results in a compound called a cerebroside. If you recall, glycosidic bonds are the covalent chemical bonds that link ring-shaped sugar molecules to other molecules, like the alpha-1,4 glycosidic linkage of maltose and the beta-1,4 glycosidic linkage of lactose. The carbohydrate portion of these compounds can be very complex. Gangliosides are examples of glycolipids with a complex carbohydrate that contains more than 3 sugars. One of them is always sialic acid. Are you starting to learn, class? Now that we are done with the last of saponifiable lipids, let us move on to, drumroll, non-saponifiable lipids. Non-saponifiable lipids are not cleaved into smaller molecules by hydrolysis due to the absence of the ester group. As what we discussed before, these ester groups are what form the long-chain carboxylic fatty acids and an alcoholic functional group to create saponifiable lipids. There are 3 classes of non-saponifiable lipids: terpenes, steroids, prostaglandins, and leukotrienes. So, can anyone repeat for me again what are the 3 classes of compounds that I just mentioned? Very good, students! So let's start with terpenes. Compounds in this class constitute what is arguably the largest and most diverse class of natural products. Majority are found only in plants. Larger and more complex terpenes occur in animals. Examples are squalene and lanosterol, made up of isoprene or isopentane units, which are common volatile organic compounds found in the atmosphere. This is an empirical feature known as the isoprene rule. But what is the isoprene rule? Does anyone have an idea on what this rule is? More importantly, do you guys even know what an isoprene is? Well, science says it is a rule that helps explain the structure of complex molecules in nature. It refers to the idea that many organic compounds, especially terpenes and steroids, are constructed from repeating units of isoprene. Simply put, natural substances are built by linking together multiple 5-carbon building blocks called isoprene units. Due to this, 5N, where N is an integer, carbons are usually found in terpenes, and they may be subdivided into the following. Now, for example, if monoterpenes had 2 isoprene units, how many carbon atoms do you think compounds under this classification have? You're right! If monoterpenes have 2 isoprene units, they have 10 carbon atoms! Remember, an isoprene unit is said to be a 5-carbon molecule. And with the 5N rule, if sesquiterpenes have 3 isoprene units, then they have, what, 3? 15 carbon atoms. If diterpenes have 4 isoprene units, they have, how many? 20 carbon atoms, and so on and so forth. Before we proceed, let's have a quick pit stop. So, for the first question, what are lipids that are not cleaved into smaller molecules by hydrolysis due to the absence of an ester group? You're right, these are non-saponifiable lipids. Next, these are a class of lipids containing carbohydrate residues and are usually the major lipids of bacterial and fungal walls. Yeah, glycolipids. I hope you guys are still following along, and let us proceed to the next type of non-saponifiable lipid, which is the what? Steroids. So, before we begin, does anyone have an idea about steroids, what they are, or what they do for our body? Well, steroids are an important class of lipids and are actually metabolic derivatives of terpenes. While they are sometimes discussed in the context of performance-enhancing drugs, they play numerous critical roles in our bodies. Steroids are typically recognized by their tetracyclic skeleton. This structure consists of 3 fused 6-membered rings and 1 5-membered ring, forming a distinctive arrangement. You'll notice that the substituents designated by R are often alkyl groups, such as methyl or ethyl groups, but they may also include other functional groups. So if I ask if there are groups can be other functional groups, what is your answer? True or false? That's right. It should be true. Always remember that. Another interesting point is the substituent at C17. Its size can vary considerably, and it's typically larger than a methyl group if it's not a functional group. Now, let's focus on our friend cholesterol. Cholesterol is the most abundant steroid in our body and plays a crucial role in cell membranes. It is also the starting material for biosynthesis of other steroids. This includes a wide range of substances such as sex hormones, progesterone, estradiol, testosterone, and aldosterone, as well as adrenal cortical hormones like aldosterone and cortisone. Additionally, cholesterol is a precursor to bile salts and vitamin D, both essential for digestion and calcium regulation. Understanding these structures help us see how slight variations can lead to vastly different functions and effects in the body. It's fascinating how the same basic scaffold can result in such diverse biological activities. So, any questions so far? Now, have you ever heard of eicosanoids? Well, eicosanoid is the collective term for oxygenated derivatives of 3 different 20-carbon fatty acids. They are signaling molecules which exert complex control over many bodily systems, mainly in inflammation or immunity, and as messengers in the central nervous system. Now, there are multiple subfamilies of eicosanoids, and 3 of these are prostaglandins, thromboxanes, and leukotrienes. These 3 are classic eicosanoids. Now, given the function of eicosanoids earlier, we can tell that they have hormone-like properties. They have specific functions, and some of these are to lower gastric secretions and stimulate uterine contractions. Were you able to write that down? Now, my question for you is, have you noticed that when you're having headaches, you sometimes take aspirin? Why do you think those work in lessening the impact of such headaches? Aspirin works by preventing the production of prostaglandins, hormone-like chemicals that play a role in pain. Therefore, their properties has an effect on the cascade of reactions associated with the hormones. Hence, our headaches tend to be slightly alleviated. Now that we know their functions or importance, let us differentiate each through their characteristics. Prostaglandins, this little structure over here, are a group of lipids with hormone-like actions that your body makes primarily at sites of tissue damage or infection. They have a short half-life and have a short duration of action. Because of this, they can only affect cells that are close by. As an example, if you cut your finger, prostaglandins will play a part by signaling platelets to form blood clots and trigger an inflammatory response. Prostaglandins also pain sensitization. Prostaglandins will act in the nerve endings, especially in the central nervous system, to promote the pain sensation and response. In the immune system, prostaglandins induce fever by acting on the hypothalamus within the brain and regulate immune cells. Prostaglandins act in the respiratory tract by regulating airway smooth muscle, promoting constriction or dilation of the airway. They regulate the GI tract, promoting contractions, and prostaglandins also decrease its stomach acid production. Within the kidneys, prostaglandins act within the vessels and the glomerulus to signal vasodilation, which is important to maintain glomerular filtration rate. In the reproductive system, they signal the uterus to contract. And lastly, they play an important role in platelet aggregation. In relation to my example, thromboxanes actually play quite the same in function. Why is that? Well, thromboxanes, which look like these, are actually substances made by platelets that cause blood clotting and constriction of blood vessels. It also encourages platelet aggregation. Allow my friend to differentiate prostaglandins and thromboxanes in platelet homeostasis.
We have two molecules, thromboxane A2 and prostacyclin. Both of them have opposing effects. So the first one, thromboxane A2, is produced by COX-1 inactivated platelets. And what this does is it promotes adherence and aggregation of circulating platelets along with vascular smooth muscle contraction. So what this does is it promotes the blood clot formation. It promotes thrombus formation. The next one we have is prostacyclin PGI2, and it's produced by COX-2 in vascular endothelial cells. Notice the difference. The thromboxane A2 is produced by platelets, and the prostacyclin PGI2 is produced in vascular endothelial cells. Prostacyclin inhibits platelet aggregation and stimulates vasodilation, and what this does is it slows down, delays, or inhibits thrombogenesis. So if you notice, thromboxane A2 and prostacyclin both have opposing effects. So the opposing effects of thromboxane A2 and prostacyclin limits the formation of thrombi to sites of vascular injury.
While prostaglandins trigger inflammatory responses, thromboxanes are the ones being secreted. Finally, we have our leukotrienes, which are lipid mediators of inflammation and chemotaxis. Taxis as in the directed migration of a cell in response to a chemical stimulus. Leukotrienes play an important role in mediating immediate hypersensitivity reactions such as those associated with asthma. Leukotriene A is the precursor to other leukotriene derivatives by epoxide opening reactions. And that ends part 6 of our lipids discussion. I hope you all learned something. Personally, I found out that glycolipids and steroids are essential molecules that control human health and metabolic processes. Glycolipids, which appear on the plasma membrane, are necessary for cell identification, signal reception, and immune reactions. Their gene abnormalities can lead to Gaucher's or Tay-Sachs disease. Fundamental physiological activities like metabolism, immunological response, and reproduction are regulated by steroids like cholesterol, as well as sex hormones such as estrogen and testosterone. Hey, but before we officially end, let's have a quick evaluation if you really learned. I'll flash the questions and you have to pick the right answers in 10 seconds. getting tired of this. Let's move on, baby, no need to reminisce. Sa akin di mo na kailangan magtaka. Nang pumunta sa iba, di mo ba gustong magpahinga? Tayo na lang nakapagod nang laging umaasa. Don't you think it's time? Come rest your heart and mind. I promise, baby. So don't you worry about a thing. Ako na lang sana piliin mo. Huwag ka na magpapalo. Sa akin na lang yung puso mo. Huwag ka na mag-iba kasi andito naman ako. Alagahin ko kahit magulang mo. Bigyan mo lang ako ng pagkakataon. Looking back, did we achieve our learning outcomes? I can see you did through the evaluation. But wait, there's more. Don't go yet because I still have some surprises for you guys. I have some assignments to give out to you today because I want to make sure you really understood my lesson. I'm getting tired of this. Thanks for watching. Please like, share, and subscribe. Don't you worry about a thing, and keep being cunts.